Okay, so yesterday we talked about RNA briefly, and we established kind of a baseline. We recapped DNA, and now we're able to, with the information that we presented on RNA, compare and contrast the two. Now, one of the biggest, or one of the most significant differences that stick out when we're comparing RNA versus DNA is that RNA is single-stranded and DNA is double-stranded. The sugars, right, are part of the nucleotide, and since both of them are made up of nucleotides, they both have a sugar. Now the sugars vary. In RNA, the sugar is called ribose. In DNA, the sugar is called deoxyribose. Your nitrogen base is, again, another component of the nucleotide. The nitrogen bases in RNA are now uracil, adenine, cytosine, and guanine. A now pairs with U. C still pairs with G. In DNA, we were using Apple's tree, car, garage, right? And so it was A with T, C, and G. Now, for transcription, we are going to change DNA into mRNA. And the reason that I use the word change is because that's what this prefix trans is referring to. The entire process, or the end result, the main goal of this is going to be to make a protein. And proteins are important, we said yesterday, because your proteins actually code for your traits that make you unique. Your hair color, your skin color, your eye color, your height, etc. All of this is already premeditated in the DNA at conception. And so now your cells have just basically undergone this process to make those proteins, right, into your physical appearance. We're going to watch this short Amino Sisters video, and it's going to set a foundation for what we're talking about today and preview what we're going to be discussing tomorrow.
After learning about DNA, have you ever wondered, how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA, well, a few exceptions, and that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters. I'll remind you to stay curious.
So, like I said, a lot of good info. It basically talked about what we're doing today, and it previews what we're going to be doing tomorrow and for the rest of the week. We're going to learn to start reading DNA, and we're going to learn to make it into proteins. And it's not as difficult as one would think. Now, the essential question, and the answer to there at the end, how does DNA play a role in all of this? Well, DNA has the blueprint. It has the instructions. It has the recipe. It has whatever it is that you want to call it. Just know that none of this would take place if it wasn't for DNA. DNA, I like to think of it like as a princess trapped in like a tower, right? You may think of Fiona. I don't care. The fact of the matter is this: it's trapped and it cannot leave. So anytime that DNA is involved in a process, the location of the process must be the nucleus. DNA is trapped in the nucleus and it cannot exit. Now, in order for us to then take that code and start making proteins out of it, what we need is this RNA, which is specifically the messenger RNA. mRNA has the ability to enter and exit, and that is why it is so important. Now, a couple of things to consider. Proteins are made of amino acids. So tomorrow we're going to be discussing amino acids, and you should not feel overwhelmed by that. And proteins are assembled at the ribosome. So ribosomes are the lean, mean protein-making machines. And there's a ton of them free-floating in the cytoplasm. If you remember your organelles, you also probably remember that those ribosomes can get attached to the endoplasmic reticulum, and it'll make it look really, really bumpy, and we call it the rough ER. Okay, so how is it that this code is going to get out to the ribosome? Well, like we said, mRNA is going to go in, copy the code, and get out. Now, a couple of reminders, because we're constantly having to differentiate between DNA and RNA. DNA is double-stranded. RNA is single-stranded. DNA has deoxyribose. RNA has ribose. DNA has thymine, RNA has uracil. There are 3 different types of RNA: mRNA, rRNA, tRNA. Focus on that first letter because it's telling you what it does. mRNA goes in and out and takes the message. rRNA makes ribosomes. Why are ribosomes important? Well, that's where proteins are made. Transfer RNA is going to deal with the amino acids, which are going to be the building blocks of the proteins, and that is the entire reason we are undergoing this process. Now, mRNA delivers the instructions for making a protein from the nucleus to the ribosome. The ribosome is free-floating out in the cytoplasm. Since, again, this is the 3rd time I've said it, since DNA cannot leave the nucleus, this process is taking place inside of the nucleus. mRNA is going to copy the code from DNA, but it's not going to be an exact copy. It is a transcribed copy because there is a change. We've changed all of our T's to U's. Once it's got the copy of the code, it takes it out of the nucleus, floats through the cytoplasm until it reaches a ribosome. Now, mRNA can't go at this alone. It needs the help of this enzyme called RNA polymerase. Now, the idea is that RNA polymerase is kind of like the muscle. It goes in there and it splits. So my DNA is a twisted ladder, right? It goes in and it splits the twisted ladder. And it's only going to do this for a very brief period of time. While RNA polymerase is separating the double helix, mRNA comes in and it starts to transcribe. So it'll run in and it'll make a copy of the code. And if DNA says A, mRNA is going to say U. DNA says C, mRNA is going to say G, et cetera, et cetera. So again, this is called transcription because trans means we're changing. C is a copy. C comes before L in the alphabet. So transcription happens before translation. And all we need is one side of the DNA because what we learned in replication is that it serves as a template. And since DNA and RNA are complementary, all that you need to understand is your base pairing rule. Again, in DNA, your base pairs were apple tree, car garage. A goes with T, C goes with G. In RNA, we now have Under Armour, car garage. U goes with A, C goes with G. You'll continue doing this base pairing rule, and there is going to be a promoter and a terminator. And this will be what allows the RNA polymerase to go in there, and it knows when to basically release the double helix and exit with a copy of the code. So there's a really good kind of video that we're going to watch here in a second. Now, when you're practicing transcribing, G goes with C, T goes with A, C goes with G, A goes with U now, G goes with C, A goes with U, T goes with A, U, G. Okay. Now, the other thing that we saw in the Amoeba Sisters video is that we're now going to start to read these in groups of 3. Those groups of 3 are called a codon. And that should not have been the first time that you read that because this was a part of your vocabulary. So you should be familiar with a codon. Now, here's the animation that I was discussing. We're going to watch it. It's about a minute long, and then we'll go ahead and we'll finish off transcription.
Transcription is the process by which RNA is assembled from a DNA template. The 2 strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the 2 strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine.
So U pairs with A.
This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
So again, RNA polymerase is the muscle. My DNA is my double helix. Everything is apple tree, car garage in DNA. Well, as RNA polymerase comes in, my twisted ladder gets separated for a small period of time. mRNA comes in, it starts making base pairs, but now my base pairs are UACG. Thymine no longer exists in RNA. And then once I have my transcribed copy of the code, it exits out of there and it heads towards the ribosome. Now, with sequencing, we are going to have these sequences that are going to either be referred to as exons or introns. And the thing about exons to remember is that they are expressed. You can see them. These are going to make proteins. Introns are the fluff. This is like when you go to English and you're writing a paper and then your teacher gets it and she starts putting lines through a bunch of sentences and she's like, this is all fluff. We don't need this. We want to get rid of the fluff. So what we actually do is that we splice the sequence together. here you have, and it's easier to visualize than it is to kind of read through the paragraph. So here's what I'm trying to get at. In orange, you have your exons. These are going to be physically expressed. These are traits that we're going to be able to see. In the purple, right, we have these introns. These are the fluff. So what we do is we actually pull the introns, and that's going to leave these gaps between my exons. We'll fill in the gaps, and then we'll basically just stitch them up together. This is called splicing, and it's done naturally. We don't actually have to go in and do anything. And so then once we have the spliced mRNA, it'll exit the nucleus because now all of these proteins that are going to be made at the ribosome. Again, because DNA cannot exit the nucleus, transcription takes place inside of the nucleus. It is going to exit, travel through the cytoplasm until it reaches a ribosome. Once it reaches the ribosome, we start the next process, which is translation. So here's an illustration of that happening. We have the double helix, we split it apart, we got the transcribed copy. It is almost identical to the DNA replication. The only difference is that no T's exist. Now we have U's instead. Okay, so when I go from DNA to mRNA, this process is called transcription. Because DNA is involved and DNA can't leave the nucleus, this takes place inside of the nucleus. mRNA to a protein, this process is called translation. Because proteins are made of amino acids assembled at the ribosome, this process must take place inside of the ribosome.